Welcome to the Monday Plenary Session. Please welcome the President of APS, Sally Miller.
Well, good morning, early birds. If the countdown didn't wake you up, you have my permission to go get more coffee right now. Welcome to this morning's plenary session, the first of our annual meeting in 2016. A special welcome to all those around the globe who are participating virtually through our live streaming video feed. We've been working throughout the year to emphasize our theme, Science to Practice. During the meeting, we'll all have many opportunities, from the pod talks to the posters, to see how we and our colleagues have been putting our science to practice, beginning with this morning's plenary session. Well, plant pathologists have been translating our science to practice and field applications for generations. John Charles Walker, APS president back in 1943, famously advised a young Paul Williams in 1961, when discussing possible directions for Paul's research, said, "Whatever direction you take, always remember to keep one foot in the furrow." This phrase has been taken well beyond to heart by a number of plant pathologists over the years, well beyond the cabbage field in Wisconsin where it was first uttered. But these words are relevant to all of us who seek through our science to make the world a better, more secure place. Nearly 50 years later, after a revolution in molecular biology that advanced the field of host pathogen interactions by leaps and bounds, Luis Sequeira, APS president in 1986, and the first editor-in-chief of MPMI, urged attendees of the 2009 ISMPMI Congress to consider the potential practical outcomes of basic research and to engage in projects that could benefit agriculture. In an interview shortly after the Congress, he emphasized the connections between basic science and downstream applications. He pointed out that he was strongly in favor of basic research in host-parasite interactions, and that you can't necessarily predict which of the discoveries are going to make it important applications in the field. But we need to think about that strongly, and actually, because of the pressing needs, need to speed up the process. We're now on the cusp of another revolution in science that promises to bring us even closer to heeding Dr. Sequeira's advice. New molecular and computational tools that J.C. Walker couldn't have dreamed of back in 1940s are now accessible and affordable to many of us. For example, some of these tools will allow us to dig deep into the mysteries of the phytobiome and ask questions that move beyond who is there to what are they doing, and how can we harness the components of the phytobiome to improve plant health and productivity. Yet the generation of new knowledge through scientific discoveries is only the beginning of the process. Translation of basic science to practical application in the field is a multi-step process that includes technical, regulatory, social, and economic factors. And that is just the beginning. Diffusion and subsequent adoption of new products and practices can be a long process that requires effective communication across multiple links in what's known as the value chain, which is a series of activities that a company or other organization performs to bring a product or service to market. The challenges of delivering science-based solutions to farmers in a way that is acceptable and usable to them is arguably greater than those of developing the solutions in the first place. Food security is a deep, recurring concern for the developing world. The theme of the 2018 International Congress of Plant Pathology is Plant Health in a Global Economy. This theme recognizes that there is still a long way to go to solve the food security crisis, and translatable new discoveries are urgently needed. While there are currently enough calories produced worldwide to meet the needs of all people, much is lost through post-harvest pathogens, pests, and waste. Further, food is not distributed in a way that meets those needs equally. Therefore, improvement of local food production systems is critical to achieve food security. And food security is not predicated only on calories, but on the types of calories consumed. Malnutrition is a severe problem. Up to 3.5 billion people are malnourished, and that has serious consequences for human development. And finally, food security is threatened by mycotoxins and human pathogens. For example, aflatoxin exposure in people is a continuing problem well documented in Kenya, but widespread in other countries as well. Numerous sporadic outbreaks of foodborne illnesses in this country have been documented and highly publicized, but this problem has not been solved. Microbial food safety is a consistent recurring problem in developing economies. Our own work in northwest Nigeria showed that high levels of coliforms and E. coli contamination are common on tomatoes during production and even more so in markets. The question has been discussed to a great extent is whether or not food production will be able to keep up with demand as the world population increases to 9 billion by 2050. The rate of yield increase in staple crops such as maize and rice is slowing, but there is room to grow in developing economies where the gap between the theoretical maximum yield and what is actually achieved is still big. Some consider the genetic potential for improvement of crops such as maize and soybeans using traditional and marker-assisted breeding techniques to be approaching the theoretical maximum. But resistance to new technologies among consumers may limit application of new approaches. Other factors include mismanagement of soils, resulting in loss of organic matter and nutrients, compaction, and other forms of degradation. Climate change is especially problematic, as in addition to increases in extreme weather events, introduces a great deal of uncertainty into production systems. And it's not just a problem in the developing world. That picture is not from the developing world, it's from my field plots in northwest Ohio last year. So climate change is affecting all of us. Climate change, as well as globalization, promotes the emergence of new pests and diseases. Finally, there are many socioeconomic factors, from unfavorable perceptions of science and technology to inadequate basic education to the cost of energy, that affect crop productivity that we as plant scientists may not be able to influence, but we can still do our part. Okay, so the point I want to make here is that there aren't any silver bullets. There are excellent technologies and crop production tactics, but regardless of application in developed or developing countries, they must be deployed within the context of production systems. Diagnostics are the critical first step in disease management, and there have and continue to be great strides in technology to improve our ability to diagnose diseases and detect pathogens in the environment and in plants. There's been widespread adoption of molecular and serological tools in plant disease diagnostic labs and field settings in developed economies. However, even the least expensive immunostrip assay is out of the reach of smallholder farmers and extension workers in the developing world. A great deal of emphasis has and will continue to be placed on improved varieties with multiple disease resistance. But we all know this is a moving target that requires both advanced molecular tools and local adaptive research to be sustainable in the long run. There's a great deal of interest in beneficial microbials. They're readily available in this country, but you can also walk into almost any pesticide dealer in a rural village in Nepal, for example, and find bags of trichoderma or Pseudomonas fluorescens for sale. There's much more that we don't know about how microbials work in the full context of production systems than we do know. But we can be optimistic that new research in the phytobiome may provide some answers. Cultural practices such as crop rotation and sanitation are key parts of production systems, but may be knowledge and labor intensive and consequently not always applied as they should be. Nonetheless, they're the foundation of healthy agroecosystems and shouldn't be neglected. And finally, pesticides are an integral part of production systems and must be continually reinvented as pathogens may become insensitive to them. And in some parts of the world, pesticides are misused, partly a problem of misdiagnosis and partly a lack of education on how and when to use them. So this morning, we are fortunate to have three outstanding scientists who will tell their stories of translating science to practice on 2 continents and different production systems revolving around maize, bananas, and potatoes. Our first presentation by Peg Redinbaugh, USDA ARS research leader and research plant molecular geneticist, also adjunct professor at The Ohio State University, will present the story of maize lethal necrosis, a disease that has recently broken out in East Africa, threatening a staple food and a key determinant of food security for smallholder farmers. She'll describe the significant progress of an international team of researchers utilizing traditional and advanced approaches to identify the disease, define epidemiological factors associated with the outbreak, and develop control strategies. Our second speaker, Dr. Leena Tripathi, is a plant biotechnologist at the International Institute of Tropical Agriculture Bioscience Center in Nairobi, Kenya. Dr. Tripathi will describe how basic and applied research approaches have contributed to the development of management options for this disease, from advanced diagnostics to cultural practices to transgenic disease-resistant plants. And finally, Linda Kinkle is our third speaker, a professor in the Department of Plant Pathology at the University of Minnesota. Her presentation will highlight metagenomic and phenotypic compositional, functional, and network data on plant microbiomes within distinct environments. She'll describe how these data can enhance our understanding of the dynamic, ecological, and evolutionary processes that influence microbiome characteristics, and how these data can guide the development of novel approaches for disease management. Really looking forward to these three talks. Before we begin, I'd like to remind everyone that we'll be holding a Q&A session following the presentations. So please tweet any questions that arise as you are listening using the hashtag Phytopath16. So now please join me in welcoming Peg Redinbaugh to the podium.
Down we go. Good morning, everyone. I'm really glad to be here, and I'd like to thank Sally for giving me the chance to share our story about the emergence of maize lethal necrosis with you. I have to remember, where'd it go? Sorry, the clicker. Oh. Got it. Okay. All right, when I say we, I certainly don't mean me. I mean all of the people, institutions, and research supporters that you see here. This work takes more than a village, but I especially want to thank all of those whose names are in bold who contributed to what I'll talk about today, and especially to thank the folks in our ARS group and our close collaborators at Ohio State. Our story starts with a seemingly instantaneous outbreak of severe symptoms and plant death on maize in the South Rift Valley of Kenya. Researchers in the region were struggling to pinpoint the cause of the problem, and CIMMYT scientists Dan Jeffers and George Mahuku were called in to work with Ann Wenge from Kenya Agriculture and Livestock Research Organization to figure out what was going on. Dan and George concluded that there was a virus involved, and Dan, who has Ohio State roots and knew about the historical expertise in maize viruses at Ohio State and ARS in Wooster, called me to see if we could help. One sec. We started with the same questions that I tell diagnostic students to ask. Where is the disease? In the South Rift Valley. When did you find it? During the short rains of 2011. That's the fall for us, those of us in the temperate zone. And during the long rains of 2012, that's the spring for us. What did you see? Well, mosaic symptoms like those caused by potiviruses, but very severe, and the plants were drying up and dying prematurely. How were the plants distributed? They were distributed throughout the field, and that's not particularly consistent with just potiviruses. Who was infected? Maize. There is no clear involvement of other crops or weeds in the region. Who were the insects in the field? Thrips. Well, that turned out to be the big clue. So I wanted to use RNA-seq or next-generation sequencing to determine what viruses were present. But every time I mention this, Dan said, "But Peg, there's thrips everywhere in the field." So we made a bargain. I would use his approach first, and if that didn't work, we'd move on to RNA-seq. And I forgot a slide. There's my clues from the field. Okay. So what was Dan talking about? Well, the only virus that we knew to be transmitted by thrips in maize was maize chlorotic mottle virus, or MCMV. So we got the samples from CIMMYT and tested them for MCMV using a tissue blot immunoassay. And this is a really quick serological test that only takes a few hours. And lo and behold, the samples lit up like a purple Christmas tree that you can see there at the top of that blot. Okay, so the symptoms seemed too severe to us for what we knew about MCMV. And we also knew that MCMV can have a synergistic interaction with potiviruses to give a disease called maize lethal necrosis, or MLN. We also knew that the potivirus, sugarcane mosaic virus, that infects maize, and I'll call it SCMV, was present in Kenya since the 1980s from work that Ray Louie had done. Sure enough, about 80% of the samples were also infected with SCMV, and nearly 70% of the samples we looked at were infected with both. So the traditional approach worked, and we had what the disease was, maize lethal necrosis. Okay. Now, SCMV and other potiviruses are pretty much found worldwide, especially SCMV. And this means that MCMV is the unique factor causing MLN. MCMV was first described from Peru in the 70s and then emerged in the middle of the United States, Kansas and Nebraska, later on in the 70s. It was then found in Argentina, Thailand, Mexico during the 80s, Hawaii in the 90s, and then there's a gap, and we have a more recent emergence in China and Kenya in 2011, and then Taiwan in 2014. Since its emergence in 2011 in Kenya, MLN has been found in Tanzania, Uganda, Rwanda, the Democratic Republic of Congo, and Ethiopia. There's also reports of symptoms looking like MLN in Burundi and South Sudan. So the affected areas where the stars are on that, clustered on that map, is actually bigger than Texas, Oklahoma, and Kansas combined. MCMV and SCMV are universally present where we see MLN symptoms, and the disease has been devastating, wiping out smallholder farmers who rely on maize for both food and income. In 2014 alone, the damage caused by MLN in Kenya was upwards of $40 million. Oops. Okay. In our initial screen, we saw 80% infection of symptomatic plants with MCMV and SCMV. So what happens when we take a broader look? In surveys from Kenya, Uganda, Tanzania, and Rwanda, we see a similar story. 50 to 75% of plants tested were positive for MCMV. SCMV was usually a bit lower, 23 to 62% here, and a strong majority of the plants infected with SCMV were also infected with MCMV. Now, I'll touch back on this in a couple slides, but we saw significant differences in the way that our antisera to SCMV, different antisera, interacted with the samples. And this suggested that there might be some substantial diversity in SCMV isolates in the region. So we did still get to use those fancy newfangled diagnostics. We did RNA-seq to look at the populations of MCMV and SCMV, In the samples we had collected from Kenya and Uganda, we're working on the samples from the other locations as we speak. And what we see is that there's little diversity in MCMV genome sequence among isolates worldwide, with all the isolates that we know about having more than 95% sequence identity. Now, that's the difference between me and a chimp, but for a virus, that's not very much. Our isolates from Kenya and Uganda were more than 99% identical to other East African isolates, and more than 98% identical to isolates from Asia. These data suggest a recent introduction of MCMV into East Africa, which was expected based on its rapid emergence. They also indicate that existing diagnostics, both serological and molecular, are probably going to be useful for detecting the virus in the region. RNA-seq gave a different story about SCMV isolates in populations. The SCMV contigs that we identified in our dataset had more than 20% sequence diversity, reflecting a significant proportion of the diversity we see for SCMV worldwide. Sequence identity within each of the 4 types of isolates that we saw, and we're calling them Rwanda, BD8, Ohio, and China-like. So within each type of sequence, the sequences were more than 90% identical. But between the isolate types, they were more like 80%. So the sequence similarity among the 4 types, so if you see on this slide, the 3 colored groups at the bottom, the Ohio isolate is in the middle of those. Those groups tend to be more virulent against known potivirus resistance genes, like the Ohio isolate is. Okay, the other thing about this sequence diversity is it suggests that our current diagnostic approaches are not really going to be adequate for detecting all of the SCMV that might be present. We knew that RNA-seq would give us a broader picture of viruses present in the Maize samples, giving us an opportunity to see viruses we weren't expecting or didn't know about. And we weren't disappointed, as plant pathologists anyway. At least 2 previously undescribed viruses were detected. One was one putative virus has genome sequence with low similarity to the Maize-infecting Polarovirus, Maize yellow dwarf virus. This was previously known as barley yellow dwarf, RPV, for you virologists out there. We also saw evidence for at least 2 other potiviruses in our samples, one having highest sequence identity to Johnson grass mosaic virus, and others similar to sorghum mosaic virus. In addition, we detected maize dwarf mosaic virus in a lot of samples that we tested from Rwanda. All of this is to say that it looks like a fairly complicated picture of what viruses are actually in the landscape infecting maize. So maize is constantly in the landscape in East Africa. It's always present. Even when it's not the major season, you'll see it outside somebody's front yard. They'll have a few plants, but it's always there. So maize is probably the most important bridge for MCMV and SCMV between seasons. Ben Lockhart, working in Kenya, also identified several crop and weedy hosts, including sorghum, millet, and foxtail. In sorghum, the MCMV infection doesn't produce any symptoms, and this makes it a stealth reservoir. You don't even know it's there. We suspected from earlier work in the US that MCMV might be transmitted through soil, like some of its cousins in the Tombas Verde. This appears to be true as the virus can be detected in the soil, as you can see on that RT-PCR slide. When George Mahuku planted maize seed into soil collected from around infected plants, he found that the virus was transmitted to almost 70% of emerging plants, and that the ability to transmit persisted for at least 50 days. So we have several reservoirs present. There's also tremendous concern that MCMV is being transmitted at a high rate through seed, and that this accounts both for reintroduction of the virus every year into endemic areas and also for long-distance movement of the disease across the region. Previous results from the US, from Stan Jensen's lab, indicated an average seed transmission rate of about 0.04% in US germplasm, with some lines having higher transmission rates than others. In Kenya, Ben found a high level of MCMV infestation of seed, and that is he could detect the virus in seed collected from infected plants and purchased in local markets. I'll add to that a recent report by Quito and co-workers that indicated a high seed transmission rates for MCMV in Ecuadorian maize. Okay, so we have lots of reservoirs. What about vectors? Well, maize thrips, which is Frankliniella williamsi, were reported by Cabañas and co-workers in 2013 as vectors of MCMV. These insects were identified in Kenya in 2008 and 2009 and have been found in every field that's affected with MLN. Sevgan Subramanian and his co-workers also showed that several other thrip species that are common in Kenya, common blossom thrips, tobacco thrips, and palm thrips, are also capable of transmitting the virus. We know from previous results of Scipnolts that coleopteran beetles like Diabrotica species and flea beetles are also capable of transmitting MCMV, but we don't really have any idea of the role of these insects in the disease in East Africa. So what do we have? We have the perfect storm for developing MLN in East Africa. The viruses MCMV and SCMV, plus several others, are constantly present in the environment with significant reservoirs in maize, other crops, weeds, seeds, and soil. And I didn't talk about this, but work done by Simmet and Calro at the screening facility in Naivasha indicated that more than 90% of pre-commercial and commercial maize hybrids were highly susceptible to MLN. Further, MCMV and SCMV vectors, thrips, and aphids are everywhere. Finally, most of the maize crop in this part of the world is under constant stress from drought and nutrient deficiency, as well as other biotic threats. So there's the bad news. So what can we do? What's the way forward? In some areas, we still need more information, as you can see from the questions that I've outlined here. But I think we're at a point where we can actually define those problems and design experiments to address them. I'll talk a little bit in a minute about looking at virus resistance to MLN viruses. But I think we have the technology available to identify and develop resistant hybrids, and my colleagues at Simmet have already released some more tolerant maize hybrids. So what are our research questions? For the pathogens, pathogen reservoirs, and vectors, we have a number of remaining questions that we need to address in order as we begin to think about long-term control of the disease. And I'm sure there's more questions than what I've outlined here. For the viruses, what are the role of SCMV and the other potivirus populations in disease development? Are some isolates or some virus species more important than others? Also, does that ludiovirus, the polarovirus, play a role in disease development? We need to get a good handle on the rates of soil and seed transmission, and we need to figure out how to control those. We need to know if there's a role for vector control in combating MLN. And if there is, can we develop these methods to resource-limited smallholder farmers in East Africa? I'd also like to know whether some of the maize hybrids coming out of programs to increase drought and nutrient tolerance or nutrient deficiency tolerance have better basal resistance to MLN. We're still missing key information needed to address the issue of bringing clean seed and soil to East African farmers. And a couple of large projects funded by Gates Foundation and USAID are targeted at gaining this information and delivering it to farmers. I'm part of a project that will be looking to develop protocols for assessing seed transmission, comparing MCMV seed transmission rates in East African and US germplasm, looking at the effects of parental status on seed transmission. In other words, when does an early infected plant produce more seed transmission than a late one? Seeing if seed treatments might mitigate seed transmission. We'll also be defining how soil transmission occurs and looking for ways to mitigate it. Of course, one major objective of this project is to bring our findings to national plant protection and extension organizations and to help improve their capacity to manage these problems. By the way, we're looking for a postdoc on this project, so please see me if you might be interested or know someone who is. Okay, so I think really in the long run, it's going to take a lot of different approaches to control the disease, some of which I've just been talking about, but a major component of this is going to be developing, delivering resistant hybrids. In a preliminary set of experiments, we identified some inbred lines that showed resistance to a US isolate of MCMV. On the left side of that table, you can see those results. Two things are of interest here. One is that many of the lines that did well against MCMV alone in the lab also did well against MLN at the screening facility in Naivasha, and that's the data on the right-hand side of the table. The other is that a number of the best lines, and they all start out with OH1VI RIL, okay, were derived from a recombinant inbred population that we had developed from a cross of OH1VI, which is an inbred line that has resistance to at least 10 viruses in 6 different virus families, and our universally susceptible inbred line, OH28. So we had previously mapped SCMV resistance and resistance to other potiviruses in this population, and we found what we expected. And on this slide, the blue bars represent where potivirus resistance is in the maize genome on chromosomes 3, 6, and 10. These loci also provided resistance to other viruses, but I'm not going to talk about that here. So when we mapped resistance to MCMV in this same population, we found loci on chromosomes 2, 3, and 10, those are the red bars on that figure, were important for resistance to MCMV. Note that the loci on chromosome 10 for MCMV and potivirus resistance overlap, and the loci on chromosome 3 are fairly close together. And so Victoria Bulugaya, who just finished her master's degree at Ohio State, tested recombinant inbred lines that carried loci from chromosomes 3, 6, and 10 to see how they responded to MLN development under inoculated and natural infection conditions. And indeed, she found that these loci are important for MLN resistance in the field and in the greenhouse. So while we still need to look at the effects of these loci in a more detailed way, it appears that lines with resistance to both viruses are going to be resistant to the disease. One thing I want to add is even though we're making progress in identifying and mapping resistance to MCMV, and I'm calling these MCMV resistant lines. The virus is present in all of the resistant lines that we've looked at so far, so they're actually more what I would call tolerant than resistant. And so we and our colleagues are continuing to screen the vast array of diverse maize germplasm to see if we can find lines where where MCMV cannot move into systemically. So, where are we? Well, we've learned a lot. We have a better understanding of the viruses and the populations that are associated with the disease, and we've identified new and potential virus reservoirs. The major vector has been identified, and we understand crop susceptibility, and we've begun to identify and develop more tolerant lines and hybrids. But we have a long way to go in order to define the roles of these newly discovered viruses, to develop better diagnostics that capture all of the problems that are out there, to deliver clean seed, clean soil, to deliver resistant hybrids and lines, and to develop a better understanding of thrips and other environmental factors that are controlling disease emergence. And with that, I will say thank you.
Thank you, thank you, Peg, for a really great presentation. I hope you're all in the audience thinking of questions that you're either tweeting or will come up to one of the microphones during the Q&A session. So now I'm very pleased to invite Dr. Leena Tripathi to the podium.
Good morning, everyone. First of all, I would like to thank Sally for inviting me and giving me this opportunity to actually share the story of the banana bacterial disease with you. So what I'm going to do is I will talk about the management strategies for this disease, which is a new emerging disease in East Africa. The first thing is to tell you guys that bananas are very important for East Africa. The world banana production is about 144 million tons, and out of that, 1/3 is grown in Africa, particularly in the East Africa is the major growing bananas. The most important part is actually the bananas are grown by the smallholder farmers, particularly women, and is not for the export. 85% of the bananas are actually consumed locally, only 15% goes into the regional and the international market. Uganda is basically the second most largest producer of banana after India, but they are the largest consumer of bananas. So there is the estimate of about 0.7 kilogram of bananas per person per day consumption. That means, people can just stay on bananas from breakfast, lunch, and dinner. It's actually almost the same like potatoes in Europe or even in US. So the dish they call matoke, which is the picture here, which is like a mashed potato. That's their main meal, and they normally eat it with a peanut sauce. So the production of banana is constrained with several biotic and abiotic factors. There are fungal diseases like black sigatoka, Fusarium wilt, viral diseases like banana streak virus, banana bunchy top virus. There are also the pests like nematodes and weevils. But the banana Xanthomonas wilt is actually coming as the most important disease for banana in East Africa. This disease is caused by Xanthomonas campestris pathovirus rem. Actually, the disease is not very new. It was initially reported in Ethiopia about 45 years old on crop called enset, which is closely related to bananas, and is actually called false bananas because it looks like banana, but then the fruits are not eatable of that crop. Then after that, it was also reported on bananas in Ethiopia, but for a long time, the disease was quite contained in Ethiopia itself. In 2001, it was first reported in 2 districts in Uganda. And after that, actually, the disease is spread very fast, and it's reported in DR Congo, Kenya, Tanzania, Rwanda, and Burundi. But within few years, actually, from the 2 districts, it has covered all the major banana growing areas in Uganda. So the symptoms, it starts with the wilting of the leaves, the premature ripening of fruits, even the fruits which looks quite normal from the outside. That means they have not much external symptom, but then the fruits has this brown scar inside, and they are rotten. But then the biggest symptom is actually the yellow ooze. You know, if you cut the pseudostem or the rachis of the plant, you can clearly see the yellow ooze. So nobody can miss out the symptoms of the disease. The disease is especially spread through the insect vector. So what happened in bananas, then the male flower, a bract falls every day, leaving a moist cushion which attracts insects. And that's how the insects takes the pathogen from the infected plant and then infect the healthy plant. But also the planting material is the another source of infection, because normally the farmers take the suckers, the baby plants, and that's how they plant in their field. So if they have the latent infection in those suckers, then they spread the disease that way. And then the contaminated farming tools are the another source of infection. The long distance infection is actually through the traded banana products, because they use the banana sheets and the leaves to wrap the banana bunches, and that's how the pathogen is spreading. The estimated losses due to this disease is actually quite high. In DR Congo, it goes above 80%. In Uganda is also more than 70% losses because of the bacterial disease. And then in other countries is in the range of 30 to 50%. There are diagnostic tools for diagnosing the pathogen, the Xanthomonas campestris pathovermucosurum. There are selective media where you can easily see the colonies, yellowish mucoid colony, which you know that is for Xanthomonas, but there are also the PCR detection for Xanthomonas. But then, you know, the farmers cannot use these technologies in the field. So we are still looking for the simple, low-cost field diagnostic tool, maybe the ELISA-based. There are some progress done on that one, but the cost is still high, which farmers in Africa cannot afford that. The Xanthomonas campestris pathovermucosurum actually has been sequenced a few years back, and it's in the type 3 secretion type of bacteria. Is closely related to the sugarcane pathogen, which is Xanthomonas vesicola vascularum. But the banana Xanthomonas actually cannot infect the sugarcane, so it's not the host for this one. All the previous work actually showed that the XCM is a highly monomorphic pathogen, but then recent work on the genome-wide sequencing reveals that there are 2 major sublineages of this one. So Ethiopia, DR Congo, and Rwanda is in one group, and Uganda, Kenya, Tanzania, and Burundi is in the second group. In terms of the susceptibility of banana cultivars, so far there is no known cultivar which has resistance against this pathogen. So that means all the cultivars are susceptible. But there are significant differences in the susceptibility among the various cultivars. So some, like the beer type of bananas are the most susceptible. Desert cultivars are also quite susceptible. And all the East African highland bananas, which are the matoke varieties, are also susceptible. But there is wild type banana, which is a diploid banana, Musa balbisiana, which is found to be resistant. Another interesting thing actually, because there are different isolates in different countries, so I asked one of my students to actually check if there is a pathogenicity difference. Then we tested like about 52 different isolates from different countries, and there is no significant difference in the pathogenicity of those isolates collected. So there are cultural practices to manage the disease, like uprooting of the disease plant. So as soon as the farmers see the disease in their field, if they can uproot either the single plant or the complete mat of bananas. It is still debate, whether the single plant uprooting is an efficient system to contain the disease. Use of the clean farming tools, the clean planting material, also debudding the male bud so that the insect cannot transmit the disease. Recently, actually, when several researchers were doing this susceptibility test of the different cultivars, they also try to find out that there are some varieties which has the persistent bract, so that means those bracts doesn't fall off, and there is I can show you the picture of that one. So this is the figure where you can see the bracts are not falling off, so that means there is no moist cushion for insects to pick the pathogen. Or so these type of varieties can escape the insect mediated transmission, but they don't have the cell mediated resistance. So that means if the farmers go and infect them by contaminated farming tools, they still get the infection. But at least with this one, that can be used as a short-term managing tool. But then there is, as I said, there is a risk when we say to the farmers to the removal of the single plant or the meth, because there are a lot of latent infection. By the time you see the symptom in the plant, we have noticed that you can already detect the pathogen in the suckers, which is the progeny plant. So is quite risky. The another thing is the adoption of the culture practices are quite low because it's quite laborious technique, and mainly the female farmers are the one which are doing the banana plantation. So that's why it's very important to develop the resistant varieties. The overall economic losses are estimated between 2 to 8 million over a decade, which is a big amount of money for Africa. And as I said that, this disease attacks all banana varieties, resulting in absolute crop loss. So that means once the disease is reported is identified in the field, then within few months, actually, you can see the whole field can be wiped off. You can see the picture, the lower picture, where this field was affected by BXW, and then there is no other remedy than to actually uproot all the plants. A crop rotation is not really practically possible in Africa, because normally the farmers have a small land, and banana is their staple food. They don't want to actually replace that crop with any other crop. So that's why the resistance has been the best and the most effective method of managing bacterial diseases. But as I said, there are no cultivated banana cultivars which has the resistance against that disease. So that means we have to develop the resistance. The conventional breeding of banana is quite complicated and quite lengthy. So that's why genetic engineering can provide alternative option to develop the resistant varieties, and that's what we are working on. So we are using in this project 2 different genes. One is called plant ferredoxin-like protein. Another is called hypersensitive response assisting protein. Both of them are cloned from sweet pepper, that's Capsicum annum. These genes were cloned by Academia Sinica, Taiwan. This HR, these genes actually work on the mechanism where the hypersensitive response is intensified because of the harpins produced by the bacteria. And these genes has been shown effective against several bacterial diseases, including even Xanthomonas. So that's why I thought that maybe this gene can provide resistance to Xanthomonas wilt in bananas as well. So we developed this project. So from the starting itself, because this is a serious problem and is threatening the livelihoods of millions of farmers in Africa, we actually we were not thinking of only research. From the starting itself, we were thinking of the product. So that's why we designed the project in that way. So we have Academia Sinica, who is the owner of these genes. Then this has the IP issues. So we involve AATF, who has negotiated the license. So we have freedom to operate. So that means we can commercialize the varieties when they are ready. And then we have a lot of capacity building on this project, because we are working very closely with the national partners in East Africa. So we are building capacity for transformation as well as the lab testing and the field testing. And then at the end, when the product is ready, we will have the a public-private partnership, so we are involving the tissue culture companies who will actually deploy the planting material for the resistant varieties. One point which I want to make sure here is, because this is a public institution who are developing this, so we don't have any IP issues or royalty thing there. So that means the first time the farmers need to purchase the tissue culture material, because banana is not the seed crop, but once they plant their material in the field, actually they are free to distribute the suckers without any extra cost to their neighboring farmers. So we developed, we have overexpressed these two genes individually in bananas. So in bananas, we use cell suspension for developing the transgenic bananas, and we have generated actually hundreds of lines and then validated them to see to check for the presence of the gene. After that, we have developed a very rapid screening method where we can inoculate into the small tissue culture plantlets the bacteria, and within 10 to 12 days the plant shows the symptoms. And so we have used that system for screening the transgenic line. The control plants normally are showing the symptoms within 14 days, and then they normally wilt completely within 1 month. But then we see a variation in the response with the different transgenic lines. So what we do is we keep these experiments for 60 days, and the lines which doesn't show the symptom for 60 days, we move them. Actually, we take the copies of those plants and pot them, and then we inoculate them in the midrib of the leaves, again the Xanthomonas. And that's what you see, that down there, the picture has a majority of the transgenic lines, because they have already come after the first test, so they have not shown any symptom. That means they were resistant, whereas the non-transgenic control plants actually completely wilted. And the point of the inoculation where the Xanthomonas was injected, you see the necrosis there, which is because of the hypersensitive response. In order to prove that, what we did was we tried to isolate the bacteria like 5 centimeter above and 5 centimeter below the inoculation point, and we couldn't detect any bacteria. So which actually clearly confirmed that, because this physical barrier was created there, and so there was no spread of the pathogen beyond the point of the inoculation. After that, we did the molecular characterization to see the copy number, because the low copy, we wanted to actually select the low copy number. We also checked for the expression of the gene using RT-PCR as well as the Western blot analysis, and also sometimes the Northern blot. Then based on our glasshouse results and the molecular analysis, we selected 65 transgenic lines. 45 of them has the HRAP gene, and 20 of them has the PFLP genes. And we planted in the confined field trial in Uganda with our collaborator, with Naro in Uganda. And our purpose was to have the disease evaluation under field condition, but also to check the agronomic performance of the plant. That means that whether they have the similar type of yield what the non-transgenic bananas of the same variety. So this is not the soil-borne disease, so that's why the field where we planted, there was no pathogen. According to the regulatory requirement, we were supposed to actually bag the flowers so insect cannot transmit the disease. And so that's why we had to do the artificial inoculation. So we inoculated the bacteria at the pre-flowering stage. And after that, we look for the symptoms. So the transgenic, majority of the transgenic plants actually showed significant resistance against the disease and didn't show any symptom at all, which was actually quite expected. That's because they've gone from the glasshouse. But all the non-transgenic control after infection showed the symptom, and they completely wilted. So we inoculated the mother plant, but we also inoculated the second generation, that is the ratoon crop. And then after the 2 infection, we tested this for the 3 different generation, but then there was no need of inoculating again because of the mother to to the progeny connection, the pathogen was already going into the sucker. And it was very interesting. If you see the mat there, which is the control, actually there was plants there, you know, which was completely wilted and dead, you know. So as soon as the small plant comes up, it gets the pathogen, because in the soil also there is pathogen, and then the plant dies off. But then on the next picture, you can see these are the transgenic bananas, and they are happily surviving there. So majority of the inoculated control non-transgenic plants actually died before producing the bunches and even the flower. Only very few plants actually showed the bunch formation. So the first slide, which is A, is the transgenic bananas. You can see they are all green and nice. So this is a variety called apple banana. So they are small bananas. They are desert type. So that's why you can see the bananas are small. It's not because of we inserted the gene there. And then the second one is the non-transgenic. So what we did was I tested for the internal symptoms. So the upper panel is for the transgenic line. You can see there is no yellow ooze in any of the fruit or the rachis or the pseudostem. The lower panel is the non-transgenic banana. So you can see that the fruits has the scars, so they are non-eatable, and the pseudostem has a lot of ooze. So from out of the 65 lines, actually we found 11 lines which showed 100% resistance in the mother crop, but also in the progenies, in the return 1 and the return 2. So for all the 3 generations, they retain 100% resistance. And that was really good news. So that means the trait is stable. Then there was also additional lines which has shown about 90% resistance in the mother crop, but then they showed the 100% resistance in the return crop. But 11 lines were a good number of lines for us to proceed further. So what we did was we tested these 11 lines for the agronomic performance, for the yield, for the plant morphology, and we found them there is no significant difference actually so in terms of the morphology, we actually tested for the number of days to flower, the leaf area, the number of functional leaves, the pseudostem gut, because that all correlates to the yield in the end. And we didn't find any significant difference among the transgenic and the non-transgenic. When I said non-transgenic, we compared the non-inoculated non-transgenic with the inoculated transgenic plants. That was our comparison. So I was also very keen to see, like, you know, in these plants, under the glasshouse condition, they were all having the 100% resistance. So what happened in the field, you know? So in the field, the conditions were very different because there were other pathogens, there were all other fungal pathogens were also there in the field. But we also wanted to see, like, you know, whether there is a difference in the expression between the lines which has 100% resistance and which has partial resistance. So we did the qRT-PCR, and we didn't find any correlation. So that means, you know, because these are the defense-related genes, you know, so the very minimal expression is actually good enough to provide the complete resistance to the plant. This was the first field trial we were doing, so we wanted to make sure that, you know, the trait is stable, the gene is stable as well. So every 6 months we were testing for the presence of the gene as well as for the expression of the gene using the PCR and the RT-PCR. So after that, we did the second field trial. We selected the 10 best lines, and we planted a second confined field trial, which also ran for almost 2 years. And we found almost the similar results. So all these 10 lines actually showed the 100% resistance, which was like obvious, you know, because they have already been tested for 3 generations. So, you know, with this slide, I want to say that, you know, developing the transgenic varieties, particularly the bananas, are actually the long term. You know, it takes about 10 to 15 years when it's done in the public institution. Maybe the private sector can be a bit quicker, you know. So we are right now on the product development. So that means what we are doing is we have now expanding into the more varieties. So we are developing 5 different varieties. 2 of them are the desert varieties, 2 matoke varieties, and 1 plantain. And, you know, the single gene based resistance has always the chances of the breaking down of the resistance. So we are also stacking the 2 genes together. And that's what we are doing. We have developed already the transgenic lines. We have selected the best lines based on the glasshouse assay, and now we are taking them to the confined field trial in Kenya in a few months' time. And our plan is actually, if everything goes well, by 2021, these varieties can be released to the farmers either in Kenya or Uganda or in both the countries. So we have also done the biosafety because that's very important. So we did using the bioinformatics approach, and we didn't find any convincing evidence to suggest that there is any potential allergenicity or the toxicity because of the HRAP or the PFLP protein. Anyways, these genes has been isolated from green pepper, which everybody use even raw. But still, we are planning to do the food safety studies as well as the nutritional quality to compare between the transgenic and the non-transgenic. The non-target effect of these two proteins has also been tested on the soil microorganism, and so far there is no evidence to say that they have any non-target effect. The socioeconomic group at IITA also did the potential adoption of the GM bananas for BXW resistance, and the good news there is actually with their survey they find out that about 65% of farmers in East Africa, they covered actually 6 countries, said that they can adopt these varieties immediately. And then remaining about 20% people say they will first see how they are performing in their neighbor field before adopting. And only about 15% says that, you know, they will not take it, which is not a big risk actually. So this study, based on the cost estimate and benefits, suggests that, you know, investment in development of the GM bananas for bacterial wilt resistance is actually not only essential, but also economical viable. And then based on this study, actually the initial adoption rate is between 20 to 70%, which will go up to 100% in like 5 to 10 years' time. So based on that, we were requested actually because to transfer this technology from banana to NSAIDs. You know, as I said in the starting of my presentation, that this disease was first reported on NSAID in Ethiopia, and more than 20 million people actually depend on this crop in Ethiopia. So we are now trying to develop the NSAID transgenic NSAID in collaboration with EIR in Ethiopia for bacterial wilt resistance. So in conclusion, I would like to say that, you know, as I said before, the banana is very important staple food crop in Africa. Farmers would not like to replace it with any other crop. BXW is threatening banana production in Eastern Central Africa for like millions of farmers. The cultural practices can contain BXW disease if all the farmers apply it, but the adoption rate is quite low. So therefore, the resistant varieties will provide a solution, and the GM bananas resistant to BXW will be ready for commercialization by 2021. So in the end, I would like to acknowledge all the groups which are, so it's NARO Uganda, they are collaborator on that project, the CARI in Kenya, and EIR in Ethiopia, and AHF are the provider for the technology. These AHF are negotiating the license for the technology, and Academia Sinica Taiwan is the owner of the technology, and the financial support from various groups. Okay, thank you very much.
Thanks, Leena, for sharing that really interesting story. And now we've got Linda Kinkle.
Thank you. And I want to thank Sally and the organizers for the opportunity to talk about our work today. The theme of the conference, as you know, is science to practice. And I suspect that everybody in this room has argued passionately that your work, specifically and uniquely, will have significant impacts on agricultural practices. We're going to make the world better. But in fact, that translation, that jump from science to practice isn't always easy, and is sometimes really, really vague and difficult to accomplish. And I think that this is especially a challenge for us who are moving aggressively into the phytobiome sphere. And as most of you are probably aware, APS has invested significant intellectual capital over the past few years in the phytobiomes. We've had a major phytobiomes initiative, a phytobiomes workshop, a roadmap, and excitingly, just this month we've launched a brand new journal, the Phytobiomes Journal. And embedded in all of these phytobiomes pieces have been a very ambitious and specific suite of proposed significant practical impacts. We've made some big promises. And I think at this point it's critical that phytobiomes researchers think hard about how it is that we effectively leap that chasm from the basic science that we're doing to practice, which is what we all argue as the reason for our research, right? And basic science in the phytobiomes research area right now, this is an exciting time to be doing basic science here. We have the capacity with omics tools to find out organisms we didn't even know existed that are living in and on plants. We can see what they're doing. We can see how they interact with insects and with the abiotic environment. Literally, we're spewing torrents of data. But just getting lots more data won't be enough. If we want to leap that chasm, I think it's critical that we have to use our data to think about these systems differently. And as we had a great phytobiomes session yesterday afternoon with some really great researchers who are starting to take this step, but I think we need to all be deliberate and actually asking how can we use our data, how could our data actually lead us to think differently to actually make that leap to practice. And today I want to take a little bit of time to tell you how we've been attempting to do this in some research in our laboratory. Our group focuses partly on pathogen-suppressive populations in soil. And one of the motivating questions for our group is what factors mediate capacities of antibiotic-producing streptomyces to suppress plant pathogens. And our work really starts actually a long time ago and well before I was born, not that I'm 30, but in the early 1940s, a potato breeding plot was established in Minnesota. And after 30 years of continuous potato monoculture, and it was a plot that had high levels of potato scab disease. But after 30 years of continuous monoculture, scab disease was gone from that plot. And as an aside, verticillium wilt was eliminated from that plot as well. And when I started at Minnesota, one of the first things I did was to begin to study that soil. And in that soil, we found lots and lots of streptomyces that were really good at inhibiting plant pathogens, including bacteria and fungi and oomycetes and nematodes. And we, in our early work, and today, in fact, we continue to do some inoculative work, but some of our earliest studies, in collaboration with Neil Anderson at Minnesota, we inoculated some of the really good antagonists into field soils, and this is one of our first field trials. Potatoes on your left were inoculated at planting in April. At harvest, you can see that inoculated potatoes had way less disease than non-inoculated potatoes. And similarly, we did work on alfalfa, and in this case, this is soil that's naturally infested with Phytophthora infestans. On your left, you see plants that just grew in the soil, and on the right, you see inoculated plants. And you see again that streptomyces can make plants really healthy. And in fact, we could spend lots and lots of time inoculating different things and finding out when they work. But the more important question seemed to be, why is that soil suppressive? Not can we get the best antagonist, but why does that soil have the activity that it does? And that's not unrelated to how it became suppressive. And so, unfortunately, we couldn't go back in time to track that soil from 1942 forward, but we were able to compare suppressive soil with an adjacent conducive soil, and we systematically said, okay, so streptomyces, maybe it has something to do with the streptomyces. Maybe suppressive soils have more streptomyces, and in fact, we found that the suppressive soils did have significantly more streptomyces. Maybe more streptomyces isn't all of it. Maybe the streptomyces in suppressive soil are better at killing. And so we looked at the streptomyces in both soils, and we found that the streptomyces in the suppressive soil, a greater proportion of them were great at killing pathogens, and also on average they had greater mean inhibition zones, which are related to how much antibiotic they can produce. And finally we said, okay, so maybe, but there's one other possible component. Maybe the streptomyces in the suppressive soil produce more different kinds of antibiotics. That is, they have a greater diversity of ways to kill pathogens. And in fact, we found that that was true as well. The Streptomyces in the suppressive soil had a higher diversity of distinct inhibitory phenotypes. And so more Streptomyces, better Streptomyces, more diverse, this is correlated with disease suppression. Now, I note we had an N of 1 in the suppressive and the conducive soil. But nonetheless, we had these three criteria, or these three findings, and we said, okay, what does this tell us? Can this tell us how that suppressive soil developed? And in fact, understanding how that suppressive soil developed, why it developed, depends upon what role antibiotics play in these communities, right? So we see a shift in antibiotic-producing communities. So why would communities shift in antibiotic activity? Why do microbes produce antibiotics? And most of you probably remember an introductory microbiology class, maybe last year, a couple years ago, or decades ago in my case, and learning that antibiotics are weapons in microbial competition. But more recently, of course, we're well aware that antibiotics also play key roles as signaling molecules and do many, many different kinds of things in microbial communities. And a critical question for us in looking at disease-suppressive soils was, is there evidence that antibiotics actually work as weapons in these soils? And in fact, in field soils, there's very little evidence for antibiotics acting as weapons. And it's critical for us to understand, is there selection for these inhibitory phenotypes? If there is, that's what we want to harness to create a disease-suppressive soil. So is there selection for antibiotic inhibitory phenotypes? Well, we decided that how we can ask that question is by looking at the communities themselves. And in particular, if there's selection for antibiotic inhibitory phenotypes in these soil communities, then populations should be better at killing isolates that they live with, that is, sympatric isolates, than isolates they've never seen before, or allopatric isolates. And so we did something that was deceptively simple, but was a ton of work. We collected Streptomyces from locations all over the world. And these two figures summarize results from about 90-odd Streptomyces isolates. And we looked at the ability of every one of these 90-odd isolates from different places around the world. We looked at their ability to kill every other isolate. And then we said, are they better at killing things they live with or things they've never seen before? And the blue bar in the top panel is a sympatric, and the yellow bar is an allopatric. Clearly, isolates are better at killing things they live with than things from other locations. And in fact, the top figure is the aggregate among all isolates. The lower figure, we've broken it down by many different locations, locations in Minnesota, in Panama, in Kansas, and other locations. Everywhere that we looked, broken down by individual locations and then obviously in aggregate, we see that isolates are better at killing things they live with. This suggests that there is clearly local selection for antibiotics as weapons. But this story was actually more interesting and complex. Among those 90 isolates, in addition to looking at how they kill one another, we looked at the nutrient use profile for every isolate using a biolog SFP2 plate. And what we found, and the data are summarized in this figure on the x-axis, we have the niche overlap between an individual pair of isolates. And on the y-axis, we have the inhibition zone, which is how good are you at killing that isolate. Each dot is a single pair. What you see is that if an isolate eats the same things that you do, you're much more likely to kill it, and to kill it bad, than if you eat different things. This was true only for sympatric isolates, isolates from the same place. Isolates from different places, in fact, you can see the R there is 0.009. There was no relationship. If you're from 10 meters away and you eat exactly what I eat, I don't care. So this suggests, again, that there is strong local selection for inhibitory phenotypes, but importantly, it also tells us that selection for these inhibitory phenotypes is not independent of nutrient use. And so if we think, in principle, if we want to try to select for these antibiotic inhibitory phenotypes, on the right-hand panel on the top, you see two species that have fairly similar nutrient use profiles. And you basically have two potential options for mediating that competitive interaction. One is you kill your competitor, but a potential alternative is that you evolve to eat different foods. And so with this idea, gosh, there's different potential outcomes, we developed a conceptual model for thinking about how might we potentially manage to produce disease-suppressive soils. And so if you look at this figure, on the far left we have the microbial community, which has a certain evolutionary potential that's defined or determined by its composition, density, diversity, and so on. Microbial populations within that community interact actively, competing in many, many different ways for many different substrates. But our data suggests that within that competitive dynamic, there's two potential coevolutionary trajectories a community might follow. One is an evolutionary arms race, leading ultimately to a high level of antagonistic activity, which is correlated with disease suppression. But an alternative, in fact, might be a coevolutionary niche displacement or niche differentiation, which will lead instead to a growth-efficient, niche-differentiated community, a bunch of friends. Now we think about this, and we ask the question, what determines whether a community might follow one trajectory or another? And this is where we begin to think about when would niche differentiation be most likely? And we think that a lot of this has to do with the food that they eat. And we have a story about cookies. So if there's a plate of Oreos, and you and I both want Oreos, I might have to kill you to get them. But if there's a plate of Oreos and Nutter Butters and Fig Newtons, in fact, I'll eat the Nutter Butters, you eat the Oreos. Neither one of us need to invest in the secondary metabolic activity. In fact, antibiotics are expensive. My fitness may be optimized not by killing you to get all the different kinds of cookies, but by specializing and avoiding that fitness cost. And so, considering this, we thought, well, it makes sense that nutrient diversity might be a key factor determining these distinct trajectories. And perhaps not coincidentally, one of the only ways that we know of for multiple different disease suppressive systems for obtaining disease suppressive soils is a long-term monoculture. And so using this model, then we actually came up with 3 specific hypotheses that seem critical to test. And they're quite simple and obvious. The first is that monocultures should be more suppressive than polycultures. Polycultures should have populations that are more niche differentiated than monocultures. And a fundamental assumption of this model is that monocultures have a less diverse carbon profile, a lower diversity of carbon than polycultures. And so we systematically went through and tested these hypotheses. And first we looked at monoculture and polyculture. We looked at Streptomyces populations in the rhizosphere of monoculture and polyculture plant communities. And this is part of a long-term experiment at the National Science Foundation LTER Cedar Creek Research Site. We focused on 4 perennial plant species. We did this for a couple reasons. One, we wanted to have plots that had not been disturbed recently, so we did have long-term coevolutionary trajectories. The other is that this is a plot in which since 1994 we've had sustained monoculture and polyculture, and we chose 4 target plant species that each have been growing since 1994 in plots of 1, 4, 8, or 16 species. We sampled the soil community. We did a tremendous amount of culturing and genomic analyses in these communities. I'm going to present just a small slice of the data today. When we looked at pathogen suppressive activity, again, we hypothesized that monocultures would have more suppressive communities. And we actually used a simple dilution plating of the soil with a pathogen overlay, and we looked at the capacity of the communities in those soil to suppress multiple plant pathogens. And here you can see the kind of data that we end up measuring, how many inhibitors, mean zone sizes of those inhibitors. Hopefully those zone sizes are clear to you. We found here, there's 2 figures here. We found, bottom line is, monocultures were significantly more inhibitory. And in fact, we've repeated this type of analysis multiple times since 2008, and we find this to be true every season in every case. On the left, we look at the y-axis is the mean proportion of inhibitors. On the x-axis, we have plant diversity level, 1 monoculture, 4, 8, or 16 species. And on the right-hand panel, we have the mean inhibition zone. So monocultures have both higher proportions of killers, and they're better at killing plant pathogens. That was good. From these same communities, we isolated, actually from the monocultures in the 16 species communities, we isolated 80 Streptomyces. We randomly isolated Streptomyces, and we looked at their nutrient use profiles for every isolate. And then here we're contrasting the percent niche overlap among all possible pairwise combinations of isolates from monoculture and from polyculture. And we see that as we predicted, niche overlap is greater in monocultures, which is to say that the polycultures are more niche differentiated. And so if you put these things together, it seems clear that the microbial species interactions within these communities are fundamental to driving the functional characteristics of the communities. That is, in the monocultures, the streptomyces are more pathogen inhibitory as a consequence of the coevolutionary escalation of interactions among streptomyces populations. In contrast, we see that the streptomyces in polyculture are more niche differentiated consistently, suggesting again a coevolutionary character displacement trajectory. What about plant diversity, right? We said monocultures and polycultures should be different. This is actually the first data that we've gotten that looks at carbon diversity in soil. It's not an easy thing to measure. We're not convinced this is perfect, but we've been doing some pyrolysis gas chromatography mass spectrometry, and these data contrast carbon compound richness for Andropogon and Lespedeza, that is big blue stem, and then a bushy-headed clover, both growing in monoculture in the red bars, polyculture in the blue bars. And in fact, we see a dramatic difference in the carbon peak diversity in monoculture versus polyculture. So putting these things together, it seems, again, critical. Microbial interactions are critical to selecting for these distinct phenotypic characteristics. But the plant is a critical driver because the plant is setting that nutrient context. The plant determines the potential for different sorts of interactions among the microbial communities in soil. And we can think about the plant in one other way that I think is critical. The plant isn't just a passive provider of nutrients. In these figures, and hopefully you've seen these network figures before, these are complicated, but the idea is simple. Every dot is an OTU, or species. A connection, a line between two dots, means that those two species are positively co-associated across samples. They're high, they have high populations, tend to have higher populations in the same rhizospheres and lower populations in the same rhizospheres. And we've clustered these, we've colored them into modules, which are collections of isolates that tend to apparently coexist across the landscape. And what we did in the upper left-hand corner, we looked at OTUs, different bacterial species that are coexisting across the Andropogon collection of rhizospheres. And we tracked those OTUs, and we said, well, gosh, if these guys hang out together, they must hang out together consistently, right? If nutrients are kind of a driver. And in fact, what we did is then we looked at whether these isolates co-associated when we're in different plant species across this prairie landscape. So then we've got Andropogon upper left, Schizocurium little blue stem upper right, and then we have the two legumes in the bottom. And you'll see that not only in the previous data we showed the plants are critical in setting a nutrient context, that is plant diversity, do you have a lot of different nutrients or not? Plant host plays a significant role in determining who you hang out with, which obviously is a critical factor in determining the potential for species interactions. And so overall, it's clear that microbiomes, soil rhizosphere microbiomes, are exquisitely adapted to the local abiotic, but especially a biotic environment, the plant community diversity, the plant host, and the suite of microbes that you interact with. Now, in fact, we've probably underestimated here a little bit. I want to spend 30 seconds. We've underestimated here a little bit of this complexity. So we said they're exquisitely adapted, but most of the data that I presented look at just pairwise interactions. A kills B, B kills C, but in fact, it's way more complicated in a really interesting and exciting way. These are 3 different communities from this same suite of plots, and each number is one Streptomyces isolate. And a red arrow indicates which other isolates a Streptomyces can kill. And so you see, in addition to having differences in how many inhibitors or killers you have in the population, the network of interactions within that community is tremendously complex and varies significantly among communities. And in fact, these 3 communities are quite different in their pathogen suppressive potential. So these networks of interactions can be even more nuanced based on networks of signaling interactions. And here we have the bottom suite of panels are for each of those same communities, the signaling interactions, in which we have a blue arrow means an isolate can enhance antibiotic production by another isolate. And a green arrow is a case when an isolate suppresses antibiotic production by another isolate. And so when we begin to think, how do we understand the functional capacity of these communities, there's a glorious complexity here that we're only scratching the surface of beginning to be able to think about. And unraveling these layers of plant community diversity, plant host, the microbiome and its structure. Unraveling these layers, it represents a tremendous challenge for us. And the easy conclusion might be, well, it's complicated. But in fact, I think that it is really complicated. But taking a step back, I think there are some important lessons that we can use to try to say, okay, how do we translate this glorious complexity to something that can lead to practice? And I think there's two, for us right now, and it's all a work in progress, for us right now we have two major take-home messages that we're trying to use to guide our work. The first is that when we're managing microbiomes, and in fact, I need to substitute that word with my apologies to Kelly Eversole. To manage phytobiomes, we need to manage species interactions, not populations, but we need to manage the interactions. And second of all, it becomes really critical that we think not only, that we differentiate what happens on a short-term response to management versus a long-term, the ecological and the evolutionary matter. And putting these together, I want to suggest that if you target species interactions and you really think about the evolutionary dynamics within the system, you actually can come up with some novel ways of approaching management. And I want you to just remember, think again, two possible trajectories. Does that have any implications? And I want to go through some possible examples now. Decades, centuries, we've used green manures. And for disease suppression, it's often about, gosh, short term, let's increase our microbial densities, we'll get some good guys in there. In fact, by understanding these phytobiomes in more detail, can we do better? Can we look at deliberately using simple or complex nutrient suites to deliberately produce highly inhibitory versus more niche differentiated communities to achieve different kinds of management outcomes? Can we look at long-term nutrient management more deliberatively to produce a specific balanced outcome? Similarly, with antagonistic inoculants, our basic traditional model has been, let's get 1 billion of this great killer out there and we'll control disease. And that's a short-term bang for the buck. But in the long term, of course, use of a single antagonistic inoculant in really high densities, there's the obvious potential for selection for resistant populations, but perhaps more importantly in the long run, if we continue to use these in soils, for instance, are we destabilizing and minimizing the potential for indigenous communities to provide significant and stable long-term disease suppression? Do we destabilize our indigenous populations? Thinking about these different trajectories, you might think about, rather than inoculating a killer, maybe what I want to inoculate is a super competitor, right? If I find somebody who can eat everything better than anybody else, let's put that guy in the soil, because in that case, in the short term, I might increase resource competition, but more importantly, can I impose enormous selection on indigenous communities, accelerate an arms race, so the indigenous communities that are highly adapted to the grower's field can produce significant suppression. Signaling inoculants, looking at the role of signaling in mediating the community functionality. We've been looking for a universal signaler. We've actually had some pretty exciting inoculative results. A signaler in the field, short term maybe upregulates antibiotic production. Long term, can we actually maximize the activities of that indigenous community in pathogen suppression by inoculation of universal signalers? And finally, synthetic communities are a big space for many of us working in the phytobiome. The short-term idea is, gosh, let's try to put together lots of different functional capacities. But in the long term, and some are doing this, there's some wonderful research trying to actually, let's not just put together the 3 things we like best. Let's try to harness the power of coevolution. Let's find things that have co-adapted and co-evolved. And will that in the long term help us achieve more stable colonization and coexistence and function? So in closing, I want to share with you 3 key points that for us, our data has forced us, 3 key ways in which our data have forced us to change how we think about phytobiomes. And 2 of these I've already told you. First, we're not managing populations. If we want to harness phytobiomes, we need to learn how to manage interactions. And second, it's really critical to distinguish those short-term ecological versus the evolutionary effects of your management system. And finally, a gap that we have in phytobiome space is we have a real paucity of conceptual and predictive frameworks, and we need to work hard to develop these because they provide the infrastructure for us to translate our science to management. In closing then, I want to challenge phytobiomes researchers and really all of us to keep our eye on the prize. We can accumulate lots and lots of data over the next years, but unless we're committed to putting the same focus and energy into changing how we think about these systems, we're going to fail. But I think we have the power in phytobiomes to be change makers, to actually harness the power of phytobiomes through new ways of thinking and achieve the practical outcomes that we've promised. Thank you.
Thanks, Linda. That was really excellent. Before we begin the Q&A session, I want to share the rules of engagement for this portion. So if you're watching the live feed, please send us your questions via Twitter using that hashtag PhytoPath16. If you're here in Tampa, and if you haven't done it already, you can tweet your question to us, or the microphones, again, are in these two aisleways over there. And so we also have JP here who will be handling our Twitter questions. So I think you'll be over at one of those microphones. So while we are getting organized here, I'd like to ask a question of the panel or the group is, what do you think in terms of conducting research, advanced research in developing countries, what do you think are one or two of the most important critical challenges to doing that kind of work in that environment? We'll start with, do you want to answer that, Peg or Leena? I'll start with Peg.
That's a really good question. And to me, I think it's consistency. There's so many really good scientists that I've interacted with in East Africa, and they seem to have trouble having consistent access to the materials, the equipment, the funding that they need to develop really robust programs.
Thank you. Do you want to add, Leena?
Yeah, I agree with Peg. And then in addition to that, actually the regulatory procedures are, means the type of, like, if you want to go into the transgenic research or that, the regulatory procedures are quite challenging because the regulators are still learning. So, you know, those can delay the procedures, and also the procurement is very challenging in East Africa still. Yeah.
Well, thank you. I see we have someone standing there. Do you have a question? Then we'll go to Twitter after that.
Yes, my name is Bob Kimbright. I'm an extension specialist at the University of Georgia, and I'd like to ask a question for our third speaker. I enjoyed all three. If my growers were in the room from Georgia today, they would think two things. One, they'd punch me and say, "You're a lot smarter than I am," which they would appreciate. But the second thing they would say, "Bob, you've always told us that crop rotation is the backbone of everything we do." And if they were able to understand anything from what you said, they would walk away saying, if we could just suck it up and endure for a period of time, we wouldn't have to worry about the diseases because the diversity would take over and we wouldn't have to fight these diseases badly. That's the message they would have taken from there. So I would like you to comment on the impact of crop rotation with the ideals that you've presented so well today.
That's actually a really great question, and the reality is growers, of course, won't wait out 15 years of crop losses to get a suppressive soil at the end of the day. And the key is where we take that information. I mean, that's the other reason why the long-term experiment was a place we could look at the monoculture and the polyculture. But the key thing, the key take-home message for us has been that we need to be more thoughtful about management of soil nutrients. We've spent over a century thinking about plant nutrition, N, P, and K, but we've not put that, even a drop of that effort into microbial nutrition. And is it, if it's a nutritional thing, if monoculture is just a surrogate for nutrition and a really focused nutrient management scheme, can we use carbon amendments in conjunction with traditional cropping to provide the benefits of a monoculture from the microbial perspective? Can we provide that kind of an end run to get the benefits without losing 15 years of production? So that would be my response.
Thank you very much.
JP, do you want to go, and then we'll get, that looks like Melanie. Okay, let's go ahead with Twitter for one, and then we'll go to the audience.
Okay, we have a question for Peg. If this, as obligate parasites, what does the complex SCMV and MCMV cause lethal symptoms? If they are obligate parasites, what do they kill their host?
So if they're obligate parasites, what are they killing?
Why?
Why are they killing? Now I see what you're saying. That a good question. It's probably kind of an accident of having a extremely susceptible host, and then you have two viruses. My hypothesis would be two viruses with the best suppressors of silencing that we know about in the plant, and so it's kind of an accident that the virus populations get out of control in the plant and it just dies. But that probably doesn't happen that frequently in a non-crop situation.
Melanie?
Question for Leena. You mentioned that you surveyed the producers to see if they'd be willing to plant the transgenic plants, but I'm wondering, did you survey the consumers to see if they would be willing to consume the transgenic fruit?
Okay, on that slide, actually, I didn't put much details. We didn't survey only the producers. So actually, the producers, as well as the consumers and the traders, the three different groups were surveyed. And based on that, this is a published report. Is it published in PLOS ONE? So if you want to go more detail, you can go to the paper. But I presented only, because the farmers are not only the producer in Africa, they are consumers as well, you know. So they belong to the both categories. And when they say they adapt for growing, that means they will consume as well. And then the consumer says, yes, definitely, because there is also the price issue. Because of the disease problem, the banana price has gone up, and so the consumers will actually benefit more than the producer if these varieties will be released and adopted. Yeah.
Oh, I see questions over on this side. Thank you.
I'm Juliet Marshall, University of Idaho. I have a question for Peg. I was thinking a lot about some of the philanthropic efforts that people have gone to. For example, there's certain wheat varieties that groups have been very interested in, and they've bought them and taken them to other places like Africa. I know situations where, to me, it seems like a well-meaning philanthropic organizations are coming in, importing high-yielding or drought-resistant plants into Africa. Can you comment on the socioeconomic implications associated with that? Because given the epidemiology of what you presented on the disease MLN, have we inadvertently created a situation by importing seed from other parts of the world that have facilitated that situation?
Okay, I never want to blame anybody for this because it's just a mire, right? But one would suspect that the virus moved around on seeds somehow. And usually when I talk to my producer stakeholders, I tell them they need to be ready that all diseases are global. Because, especially with maize and some of the major grain crops, seed moves all over the world all the time. I think what we need is better ways to anticipate that problems are going to arise and be sure and cut it off before it moves. A lot of times what's moving is breeder germplasm and not so much hybrids or anything that goes to a consumer, because, for example, I talked about those lines that we developed. They do pitifully in Africa. They have no adaptation at all. So what we would be delivering is more, what are the genes that you need to incorporate into your local germplasm to get what you need?
I think given the Phytobiome Initiative, I think we have a better understanding, or could have a better understanding of a greater picture of susceptibility and diseases as we move some of the germplasm around.
Thank you. Next question.
Catherine Jones, Florida State University. I have a question about the phytobiomes. When there's a really destabilizing event, like acquisition of resistance by the pathogen, or even in the case of Streptomyces, ability of the pathogen, acquisition of genes for producing new antibiotics, and disease takes off, do you have sort of an accelerating event where, as plants are dying, the carbon profile changes, and then the suppressive ability of the soil gets lost, and so?
That's a really great question, and for our system, we haven't seen pathogen resistance in a field setting occur, and we think it's because there's just so many different antibiotics being produced that it's a virtually insoluble problem. We have looked hard in culture. We have gotten some pathogens that have a modest level of resistance to the antibiotics, but they tend to, at the same time, significantly lose their virulence. So there does appear to be some fitness trade-offs there. I also want to apologize to collaborators in the room. I was so happy to be done. I did not show my acknowledgement slide. JP Dundor-Arias, Lindsay Hansen, Dan Schlatter are all here and contributed significantly to the work, and thank you, USDA, for the money, and NSF. Thank you. Sorry.
Thanks. I think I will just go to Twitter, the Twitterverse for one question, and then we'll go to the left and then to the right. Okay? Oh, okay. We'll do the Twitterverse next.
Yeah,
go ahead.
The question is for Dr. Kinkle again. You gave an example of potato monoculture, but there are other places where, for example, marrow plots and I think Northampton, where they have been growing either maize or wheat for years. Do you have any idea how, or is there any work on those?
We're currently studying a collection of 5 different species that have been grown in monoculture since 1959, and that work is at its very infancy, so I can't tell you a lot about it yet. But one reason, I mean, I've been challenged before, people have said, well, not all monocultures, certainly. And I think that's true. It's unlikely that every monoculture will achieve this sort of glorious golden outcome. But I think that understanding those carbon profiles might be a really important part of the puzzle of understanding when they may be most effective.
Okay, JP?
Yeah, this question is for Peg and Leena. Are the governments in East Africa behind you to support the 2 research projects to deal with the 2 different diseases? Can you hear me?
Yes, for the most part. I mean, the governments and the researchers in the region are, they're proud of what they do. They're proud of what they're capable of, and they want to do everything that they can on their own. But they appreciate the collaboration. I mean, I don't look at it as I come in and save the day. We're working with people there who really understand what's going on in the ground. Trust me, I no clue the first time you're there. And, you know, I'm used to corn growing in rows and being 12 feet tall. It's much different there. And so, yes, they are supportive, especially as if you come in as collaborators.
So my response is the same. So the government is quite supportive of the research what we are doing for the Xanthomonas wilt in bananas there. So the national institutions are our collaborators both in Uganda and in Kenya. But also we have actually quite higher support even from the regulators, because we need the regulatory approval on each procedure. So we keep them informed, and they are also very supportive. Actually, by last slide, after acknowledgement, I didn't spend any time on that because I thought people will read it themselves. That's where actually we have support even from the president of Uganda, who says that, you know, these varieties are very important for Uganda and they are about to be released. So we have the support actually at very higher level in the country.
I think sometimes people don't understand how important food is as a cultural sort of staple as well as a food staple, so I can understand that. It's great. We have a question over here.
Yes, my name is Paul, and I'm from the University of Arizona. So the last person who asked the question actually grabbed it out of my mouth. But still, along those lines, I know that GMO is a very contentious topic in many parts of Africa. And you have a good technology which potentially could solve bacterial wilt. And so this is a good example of a science that we can practice, that we need to put in the hands of practitioners. But on the politics side, is there, in these places you work in, is there a bill or an actual act of parliament that proves this support these governments have towards genetically modified organisms?
Okay, in Uganda, actually there is bill which allows you to do the research up to the field conditions, so you are allowed to do the field trials, confined field trials. There is no law yet, so that means we cannot commercialize the GM bananas yet until they have the law. But they are in the process, so the law will be in place pretty soon. That's what we all are quite optimistic. In Kenya, actually, Kenya has both the bill and the law. So, you know, if people like, they can release the variety in Kenya.
Very good. Another question?
Thank you. I wondered, since the maize lethal necrosis has been around in Nebraska, I think you said, for a long time, why it didn't blow up there, but then it did in East Africa?
That's a good question, and one I get asked all the time in East Africa. So usually, to me, it boils down to two reasons. One is winter, which means we have a 6-month maize-free period in this part of the country. And the other is there's not a good vector. The coleopteran, the beetles, maybe move the disease around within fields in Nebraska, but it just doesn't spread very fast. There's no thrips in the region.
Thank you.
Thanks, Peg. Let's go over to here, to this side.
Yeah, Saeed Mashahid from NC State. My first question is for Leena. I heard you referring to the acceptance for the GMOs produced. Do you think that it is a true acceptance or a lack of options? Especially in this area where we have, as you said, banana and maize, and if we add wheat with UG99 and the rapid evolution for everything mainly on the plate, which is mainly because of having the crops all year around. So is it a lack of options, and is there anything that we can do for having those crops all year around to prevent the pathogen from having that step ahead all the time? Thanks.
Okay, actually, it's a really nice question. So I will not say that there are a lack of options, because in terms of the crops, there are a lot of options farmers have in Uganda, particularly in Uganda. But then, they really want bananas because that's their staple. And as I said during my presentation, that the cultural practices, that's the short-term option, and if the farmers can, they are actually, some of the farmers are applying those technologies, But because not all the farmers are applying those technology, that means, still the problem is still there. So that's why the farmers are looking for the best options, which are the resistant varieties. The farmers actually doesn't understand much the science behind these resistant varieties. What they want are the resistant varieties. And that's why they said, as soon as the varieties are released, they will adopt the varieties. So that's my answer to that. I don't know whether I've answered what you were expecting for your question. Yeah.
Thank you, Leena. We're getting close to the end, but we have two people. We'll start over here, and then we'll go over to Gary, I think I see. And then if there's a burning question on Twitter, we'll do that. So try to finish up. Okay, thank you.
Debbie Samek, USDA ARS. Another question for Leena. How universal are these transgenes for controlling other diseases like bacterial wilt from Ralstonia or Fusarium wilt, or even viruses?
So for Fusarium, actually, we tested these bananas because I thought that it can provide the broad spectrum resistance, so it can provide the resistance to the fungus. But we didn't find any immunity type of thing. We find little bit some percentage of resistance for Fusarium, but under the field condition, because these varieties, what I was showing in my presentation, is actually quite susceptible to Fusarium, and I see in the field actually they are getting infections. So definitely is not providing resistance to Fusarium. In Ralstonia actually is reported on some of the crops, but we haven't tested on bananas because Ralstonia that is not present in East Africa. So, and we have license, so we have negotiated the license and we have freedom to operate only in Africa. So I have not able to send my bananas somewhere else to test for the Ralstonia. Maybe we can do that at some point. Okay.
Gary.
Yes, I guess my question is directed to Linda. I was really excited about your work, and especially with the Phytobiomes Initiative. I've been past 9 years with the University of Florida evaluating a number of biological control agents, and I can say that I've been somewhat disappointed in a lot of the efficacy I see from these products. And I was very interested about your statement that you made in regards that potentially if we put too much of something in the soil, we could disrupt the community. But I kind of wonder if it's the other way around, that the reason why these products aren't as effective is because they can't compete with the community that's already there. Can you speak a little bit maybe about this?
I think there's some beautiful work that's showing that many biocontrol agents don't effectively colonize. And so you sort of have a catch-22. You don't get control because they don't colonize, or you get control and you mess up your indigenous community. And I'm not sure which one is the better outcome in that case. We did spend a season trying to look at whether or not the success of an inoculant was related at all to the potential for the indigenous community to kill the inoculant, right? Because all these inoculants are also likely sensitive to some populations in the soil. We didn't see a strong signal there, but it was only a single season. But it's a great question.
So just as